So what do you know about the fertilization process? What is the basic definition for fertilization? Fusion of male and female gametes. Okay. What are the steps involved in the fertilization process? Zygote formation. Okay. Blastocyst. Okay. Implantation. Fine. Gestation. Okay. Okay. What are the hormones or enzymes involved in the process of fertilization? And okay. Can you name the structure in this diagram? Okay, uterus, fallopian tube, and this one. This is cervix. This part is cervix. This part I'm asking. This is called as the fornix of vagina. Okay. Then this part what you are seeing is the cervical canal. Cervical. Okay. And this part is called as the uterus, followed by fallopian tube or uterine tube. Okay. So what are the parts of uterine tube? How many parts are there? 3 parts. What are those? Okay. What is this first part called? This is called as isthmus. Okay. The next large part is called as ampulla, followed by infundibulum. Okay. Then we have this is called as fimbriae. It is also part of your uterine tube. Okay. And later we can see the presence of ovary. Okay. So what is the name of the muscle that covers your uterus part is called as? It is called as this is called as myometrium. Okay. These are the few important parts you should remember. Okay. Once the copulation happens, there will be an ejaculation in the vagina of the female reproductive tract. Okay. So your ejaculation fluid includes ejaculation fluid. What are the components of ejaculation fluid? Okay. Mainly it is semen, right? So what are the components of semen? Plasma. It includes first and foremost thing sperm plus water. A small quantity or a small amount of water. Okay. Second thing, fructose. Fructose plus citrate. Citric. Okay. What about the third component? It includes secretions, like, can you name? Okay. Secretion from the bulbourethral gland, prostate gland, and also from the seminiferous tubules. Okay, fine. The next component is fibrinogen. Okay, fibrinogen plus. What is the another one component? Fibrinogen plus. Next one is prostaglandins. Prostaglandins. The other two important things to remember is mucus, and the last one is buffers. Buffer. Okay. So your semen includes the sperm plus small amount of water, fructose plus citric. What is the function of fructose and citric in the ejaculation fluid? It provides nutrition. Okay, it provides nutrition. Next, the next is fibrinogen plus. What is the important thing here? Fibrinogen plus. Fibrinogen plus okay, fibrinolysin. Include one more, fibrinogen plus fibrolysin plus coagulase. Important one, coagulase. Okay, all these three components helps in the binding and attachment. Okay, it helps in the binding and attachment process. Okay, the next one is prostaglandin. What does the prostaglandin do? What is the importance of prostaglandin? It helps in the contraction of the uterus. It helps in the contraction of the uterus. Okay, the next one is mucus. Mucus acts as a lubricant. Okay, it acts as a lubricant. What is the function of lubricants? It avoids the friction and helps in the smooth movement. Okay, that is the function of the lubricants. And the last one is buffer. What is the purpose of buffer? Maintenance of pH. Maintenance of pH. These are the functions of different components present in the semen. Any doubts? So fructose and citric helps in providing the required nutrition, followed by fibrinogen, fibrolysin plus coagulase helps in the attachment and binding of the sperms into the wall of vagina. The last fourth one is prostaglandin. It helps in the contraction of the uterus. Okay, followed by mucus. Mucus acts as a lubricant. It avoids any friction and helps in the easy or smooth movement of the sperms. Okay, followed by buffer. It maintains the pH inside the uterus and make the smooth movement of the, or more favorable movement for the sperm to swim. Okay, any doubts? Shall I clean? Others? Okay. Okay. Once the ejaculation takes place after the copulation, around 2 to 5 ml of semen will enter into the vaginal part or into the female reproductive tract. Okay, so this 2 to 5 ml of semen includes how many sperm? What is the count of sperm? It includes around 200 to 400 millions of sperms. Okay, so once the copulation takes place and the ejaculation happens, it will enter into the vagina of the female. Okay, now only a very few sperm will move into your uterus part. The other sperm will get attached to the wall of the vagina and later it will degenerate. And this attachment will be done by your coagulase and fibrinogen components. Okay, once the semen fluid flows into the uterus part, the prostaglandins will come and attach into the walls of the uterus like this. Okay, so now this prostaglandins will help in the contraction of the uterus. Contraction of uterus. Contraction happens in this direction and also in upward direction. As a result, the sperm enters into the cervical canal will easily flow towards the upward direction and then it will pass in the uterine tube. Okay, once it enters the uterine tube, what happens? What happens in the uterine tube? We have ovary here. Okay, the rupture of ovary takes place and the secondary oocyte will enter into the uterine tube. Now with the help of fimbriae or small cilia-like structure present on the wall of the uterine tube, this oocyte will travel to your ampulla region. Okay, from here it will travel to the ampulla region. By that time the sperm will also travel and it will reach to the ampulla part. Okay, here in the ampulla region, the fusion will happen. Okay, this is the ampulla part. In the ampulla region, the fusion of male and female gamete will take place. Okay, there are chances of your sperm coming until the infundibulum part, or there are chances of your ovary traveling into the isthmus part also. Such kind of fertilization are considered as abnormal site of fertilization. Okay, only if the fertilization takes place in the ampulla region, it is called as the normal site or the proper site of fertilization. This is how in general the fertilization process takes place. Any doubts? Any doubts? No doubts? Okay. Now we will see what are the changes that happens in the male and female gametes before the fertilization. Okay, because male and female gametes are completely two different structures. What is the difference between male and female gametes? The major differences? Male gamete is motile whereas female gamete is immotile. Okay, male gamete is smaller in size and female gamete is larger in size. In the male gamete we can see very less cytoplasm presence, but in female gametes we can see more cytoplasm presence. These are some three basic and important differences you should remember. Okay, so what are the changes that takes place in male gametes? Shall I clean this? Oh. What are the changes that takes place in the male gametes before the fertilization? So the process of fusion of male and female gamete to form a single cell structure called zygote is the old process of fertilization. Okay, and it happens or takes place within the 24 hours of ovulation, and it happens in the most dilated part, that is the ampulla. Okay, these are the results of the fertilization, and these are the steps we should concentrate while reading about the fertilization process. Okay, the first one is approximation of gametes, then followed by fusion of gametes and the results of fertilization. I'll discuss about A and B, C you should discuss. Okay, I'll give some time, you can study from your textbook and you can discuss about the results of fertilization. Okay, it's an easy topic. First we'll start with the male gamete. What is the structure of male gamete? Okay, we have head, middle piece and tail part, right? Okay, so this is the head part, and then we have middle piece, and then we have the tail part. In the middle piece we can see the presence of spiral mitochondria, and in the remaining part we can see the axial filament. Axial filament helps in the movement of the sperm, and the mitochondria will provide the required energy for that movement. Okay, what about the head part? We can see the presence of acrosomal cap, then we have the, what is this? Nucleus. Okay, so this is the plasma membrane. Then this part is called the outer acrosomal membrane, okay, which is closer to plasma membrane is called the outer acrosomal membrane, and this part is called the acrosomal cap part, which includes what enzymes? Hyaluronidase, acrosin plus trypsin. Okay, write down trypsin, Trypsin, trypsin. It is a protein digesting enzyme present in the acrosomal cap part. Okay, and this part is called the inner acrosomal membrane followed by nucleus. Outer acrosomal membrane, acrosomal cap part, where we can see the presence of different enzymes, followed by inner acrosomal membrane, which is closer towards the nuclear side, is called as the inner acrosomal membrane. Then we have nucleus. Okay. Next, what about the female gamete structure? Enzymes. Hyaluronidase, acrosin plus trypsin. Hyaluronidase and acrosin I mentioned in previous class only. Trypsin is a protein digesting enzyme. Okay. What about the structure of female gamete? This is the oocyte. Okay. Inside the oocyte we have nucleus, which is carrying the DNA, 23 numbers of chromosomes. Okay. Here this nucleus is also carrying 23 numbers of chromosomes. This is nucleus. Okay, this is oocyte, followed by oocyte is covered by or lined by. I'll keep it simple. Right now I'll mention it as plasma membrane. Okay. Since I mentioned this as nucleus, consider this as a cell. It is covered by plasma membrane. Okay. After the plasma membrane, we have, I'll draw only the half part. Okay. This side I will not draw, only the other side I am going to draw the structures. What is this layer? This is zona pellucida. Okay. After the zona pellucida, we have corona radiata. Okay, I'll write corona radiata like this. Okay, now tell me what is corona radiata is made up of. What is corona radiata? After the zona pellucida we have corona radiata. So what is corona radiata made up of? What are the types of cells, or what are the types of different structures, components we can see in the, at least why the name corona radiata? Why it is called as corona radiata? Corona radiata is nothing but a layer of your follicular cells. These are nothing but follicular cells, which is arranged in a radial manner. That's it. Okay. So between these follicular layers you can see some structure or some fluid like this. Okay, this is nothing but the hyaluronic acid. Hyaluronic acid. This hyaluronic acid will form a jelly-like substance that will hold all the follicular cells and layers together. Okay, because your oocyte is not only covered by single layer of follicular cells. At the end of the maturation process, the oocyte is covered by 5 to 6 layers of follicular cells, and those layers are called as corona radiata. Okay, so all these layers are together held by the hyaluronic acid as a jelly-like substance. Okay, so once the maturation fertilization process happens, now imagine the process is taking place in the ampulla of the intrauterine or in the uterine tube. Okay, now the male gamete will come and it will try to fuse with the female gamete. Okay, so now what happens here? What are the changes the male gamete should do before it entering? The head will get, okay, the head will pierce the zona pellucida layer. Okay,
The presence of acrosome will negatively others.
Okay, the presence of acrosome will give negative ions or negative energy to the other structures present nearby. Okay, any other? The acrosome cap will rupture. What happens if the acrosome cap ruptures? Okay, the content present inside the acrosomal cap will come outside. Okay. Anything else? Others? Oh yeah, right. The ovum will make sure that only one sperm will enter for the fertilization process. Okay, next. Whatever you all said is right, but there is two important headings to study this entire process. It is called capacitation. Remember? Capacitation and acrosomal reaction. The first step is capacitation and the second step is acrosomal reaction. Okay, first we will see what happens in the capacitation. What do you mean by capacitation? Capacitation is nothing but the maturation of your spermatozoa. The process of maturation of spermatozoa is called the capacitation. So when imagine this has the spermatozoa or the sperm. Okay, when it enters into the ampulla region, it will be having lot of proteins attached to it. Lot of proteins attached at the wall of the plasma membrane. Okay, along with the proteins we can also see the presence of cholesterols. Okay, so now this structure should undergo capacitation process, that means maturation process. Okay, once the maturation of this spermatozoa happens, before going for the fertilization process, it is going to lose few proteins present on it and also all the cholesterol part present on the head region. Okay, it will lose all the no cholesterol. Okay, after the maturation, and it will also lose the proteins that are not required during the process of fertilization. Only the proteins which are required for the part of fertilization will be kept, other proteins will be removed. So now the sperm will become more active. This condition of sperm is called hyperactive part. Okay, then it will become more motile. It will show more mobility or movement. Okay, this process is called the capacitation. Once this capacitation happens, then this structure is going to fuse with your, it's not zona pellucida, first with the follicular layer, or you can say corona radiata part. Okay, remember, not this structure is going to fuse with the corona radiata. This structure, that is the mature spermatozoa, is going to fuse with the follicular cell layer. Okay, any doubt in the capacitation? No doubts? So this you mark it as first step, that is the capacitation. From here you mark it as second step, acrosomal reaction. Okay, once your sperm touch or interact with the follicular layer, it will release the hyaluronidase enzymes, okay, which is present in the acrosomal part. Okay, so if it should release the hyaluronidase enzyme, what are the structures should be removed from here? First, the plasma membrane should rupture. Okay, it should open. Then a part of outer acrosomal membrane should also get open. Okay, so in this region, now there will be small opening. Okay, it will release the hyaluronidase enzyme into your corona radiata layer. Okay, as a result, the hyaluronic acid present in the corona radiata cell structure will get loose now. Okay, it will lose its jelly like substance and all the follicular layers will start getting separating from each other. As a result, this entire part is going to open. Okay, so now your sperm can move until your zona pellucida part. Okay, any doubts? Remember, your acrosome part have hyaluronidase enzyme, right? As soon as the sperm touches the follicular cell layer, the hyaluronidase enzyme will be released outside. Okay, as a result, it will interact with the hyaluronic acid and it will remove the jelly-like substance present between the follicular cell layers. So now the follicular cell layer, which is present as a group of 4 to 5 cell layers group, will start getting separated and also we can see gap between the cell. As a result, your sperm will move towards the zona pellucida layer. Is it clear now? Okay. What happens in the zona pellucida part? The sperm pierces the zona pellucida, but how? This is the second step, okay? The hyaluronidase enzyme is the second step. Next, in the third step, your zona pellucida will have 3 important proteins. What are those? Zona pellucida have 3 proteins. It is called as ZP1, ZP2 and ZP3. Okay. During the time of fertilization, ZP3 will help us. Okay. So on the walls of zona pellucida, we can see the presence of ZP3 proteins. This is the ZP3 protein. Okay, now the receptors present in your sperm will go and attach with this protein. Okay, it has a, yes, it will go and attach or interact with the ZP3 protein. Okay, so if this reaction should happen, the acrosomal cap and also the inner acrosomal membrane should rupture. Okay, now we are present only with the nucleus. As soon as the ZP3 and the receptors of the sperm get attached, your sperm will receive a signal to engulf the or digest the zona pellucida layer. Okay, because of the enzyme present, which one? Trypsin. I mentioned trypsin as a protein digesting enzyme and also one more enzyme is acrosin. Hyaluronidase is already used in the previous part. So now we are left with 2 more enzymes. Those will help to digest the zona pellucida layer. Okay, this is the third step. So now your sperm has almost reached the plasma membrane layer. What happens in the plasma membrane layer? Now imagine this is the plasma membrane layer, then we have the oocyte and nucleus. Your sperm is only left with nucleus part. And a part of inner acrosomal membrane. Okay, what happens now? How this pronucleus part will enter into the oocyte? Again, your plasma membrane also contains some proteins, okay? These proteins will bind with the inner acrosomal membrane, okay? As a result, the Na plus ions will enter into your, the positive ions, Na plus ions will enter into your sperm region, okay? This will activate the sperm and it will form a channels like this in your plasma membrane. What are these channels called? These are called as ion channels. Ion channels. Okay. At this point, this is the sperm that is going to fertilize the female ovum. What happens to the other sperms? What happens to the other sperms? It degenerates, okay. Anything else? There is something called fast block of polyspermy and slow block of polyspermy. It is important MCQ question. Fast block of polyspermy, slow block of polyspermy. Once the formation of ion channels takes place, the Na plus ions will also enter into the cytoplasm of the female gamete, okay. The smooth endoplasmic reticulum already present inside the female gamete will also release the Ca plus ions, okay. So now there will be a positive gradient inside the female gamete and outside we have negative gradient. So this positive and negative gradient interaction will stop other sperms to enter into the ovum, okay. This condition is called as the fast block polyspermy. Understood? No? Okay. Okay, I'll explain once again. Remember the Na plus ions. Once after the formation of ion channel takes place, the Na plus ions will enter into the female gamete, okay. Your female gamete also includes smooth endoplasmic reticulum. This smooth endoplasmic reticulum will produce the Ca plus and Ca plus ions. So now positive and positive ions will cause the positive gradient inside the female gamete, okay, and there will be negative gradient in the outer aspect, okay. So this positive and negative interaction will equalize the whole condition and it will not allow any other sperm to enter into the female gamete after one sperm enters, okay. This whole condition is called fast block of polyspermy. Then what do you mean by slow block of polyspermy? Okay, take a assignment question. Write a short note on slow block of polyspermy. 5 marks question. Write a short note on slow block of polyspermy. It is similar to your fast block only, okay. Write it as a 5 marks question with this diagram. No need to draw this diagram, just not exactly opposite, it involves one more structure. You have to figure out which structure. If you know which structure, it will form blockage like this, that's it, okay. So this blockage will stop the entry of other sperms. You just have to figure out which structure is forming this block in the slow block, that's it, okay. Try to draw this diagram only. So now the male pronucleus will enter into the cytoplasm of the female gamete. Okay, so now after this, one last step is there. What is going to happen in the last step? Remember, the general definition will always say the fusion of male and female gamete is called as the fertilization, or the formation of zygote is by the fusion of male and female gametes, right? But if you have to understand the whole concept properly, it is actually the male pronucleus and female pronucleus which is getting fused with one another to form the zygote. Okay, so now tell me how this pronucleus is going to fuse with female pronucleus. Nuclei or nucleus? So now there is male pronuclei, okay, which is carrying n number of chromosomes, that means haploid number of chromosomes. Here we have the female pronuclei, but it is arrested in which phase? It is arrested in the second stage of what? It is arrested in the prophase of meiotic second division. Okay, so now the Ca plus ions what produced by your SER will help the female pronucleus to go under the cell division and complete the whole cell division. Okay, as a result, by the end we are going to get one functional ovum plus 3 polar bodies. Remember the oogenesis? So now this functional ovum will be recognized by the sperm, and these two are going to fuse with one another to form the gamete, zygote. Is it clear? Okay. So this is the male pronuclei, okay, and we have female pronuclei. This is the female pronuclei, which is also carrying haploid number of chromosomes. And this cell is it got arrested in the prophase of meiotic second division. Okay, remember your smooth endoplasmic reticulum is producing Ca plus ions. These Ca plus ions will help this one to go under the cell division and complete the whole process. At the end of the process, if you remember the oogenesis, we will get only one functional ovum and 3 polar bodies which are non-functional due to the lack of cytoplasm. Okay, so that functional ovum, this is the functional ovum, it will be recognized by the sperm, and these two will get fused with one another to form the zygote. Then the zygote will be diploid number. It carries diploid number of chromosomes, 23 pairs or 46 chromosomes. This is how the whole process of fertilization takes place. That is approximation of gametes. Approximation of gametes is nothing but capacitation and acrosomal reaction. Then the fusion of gametes from the acrosomal reaction to this part. Whatever I have showed, no? Until this part, fusion of gametes will come. Results of fertilization you can discuss. Any doubts? Any doubts in the capacitation? Any doubts in the acrosomal reaction? Any doubts in the fusion of gametes? Any doubts in the fusion of gametes? No doubts? Sure? Everyone? If you didn't understood anything, you can raise your hand. Okay. Fine. You can check the diagrams. Whatever notes you have taken, you can compare the structures with that. Okay. If you haven't taken any notes, you just try to revise the topics or points, whatever I have discussed. This is the very first diagram I have written. You can check for all the female genital tract structures. Okay, just a minute. Then you can see the transportation of the sperm, starting from vaginal part to the ampulla part. And the release of ovary, that is showing the rupture part, and then it is releasing into your uterine tube. Any doubts? No doubts? Okay. Okay. This one I particularly added to show the rupture of, this is the ovary part, okay. Whatever you are seeing here, ovary, it includes different types of follicles. Some are in the initial phase, some are already in the maturation phase, okay. Now the rupture of ovary is taking place and it is releasing the secondary oocyte into your uterine tube part, okay. And the walls have the cilia. It will helps in the movement of the secondary oocyte from the ampulla or the fimbriae region to the ampulla part, okay. Any doubts? No doubts? Okay. From here you can check the diagram, you can compare the points or whatever the notes you have taken. If you have any doubts, we can discuss. So what is happening in this picture or in this diagram? Any doubts? No doubts? Which part? Polar body. Here what they have done in this picture is they are trying to show all the stages together. If you see the very first stage, they are trying to show the corona radiata part where the sperm is entering. After that, they have even showed the sperm entering into your zona pellucida part. And then if you come to the cytoplasm of the oocyte, this part, here they have already showing the divisions of the division of your oocyte, okay. So they are showing which phase is this? Prophase, okay. So they have tried to show the first polar body. When the prophase happens, there will be first polar body. By the end of the process, we will get total 3 polar bodies, okay. They have tried to show all the stages together here, okay. In your exam, you are supposed to write this diagram only. You won't get enough time to draw each and every diagram what I have shown. So this is the one right diagram to draw for the fertilization process, okay. Next, this is like step by step they have tried to show. You can see. F and G we are going to discuss in the next session, okay. Just try to observe the pictures right now, but E part we have already discussed few points. F and G we are going to discuss in the coming classes. Any doubts? No doubts? Can you identify this picture? What is this? Spermato. What is this process called? Any name? Yeah, it will degenerate. See, all the sperms, there are like around 300 to 400 sperms will enter into the female genital tract at the beginning, right? But the time it reaches to your intrauterine, stop talking. By the time it reaches to your intrauterine tube, the number will go down to 300 to 400. Okay, so whatever the remaining sperm present in the female genital tract will lose its capability for the movement, and later it is considered, later they'll say like it will die inside the female genital tract and it is going to degenerate. Once anything that degenerates in the body, it will be considered as a foreign body and it will be removed through the waste product. Okay, the same happens with the polar body also. Polar body is basically non-functional cell body, right? The non-functional cell body means less cytoplasm, no proper cell organelles are present. So initial and gradually it is also going to degenerate and it will also be considered as a foreign particle in the body, in the female body. Okay, any doubts? What is this process called? It is already studied, right? Blastocyst. Gastrulation, okay. What happens in the gastrulation? See, once the formation of gamete takes place, from there we are going to study as the first week of development. Okay, until here it is considered as the general part. From now onwards we are going to study it in the week wise. First week development, second week development and third week development, like that from the next class. Okay, what is this diagram? These diagrams will be discussed in next class, so that I am showing it now only, so that you will have some basic idea about the diagrams. This diagram or picture actually shows the formation of blastocyst. This last structure what you are seeing, no, this is nothing but your blastocyst structure. What is the significance of blastocyst? It gets implanted into intrauterine or into your uterine wall. Okay, fine. Any other significance? Formation or completion of blastocyst will indicates to the formation of 3 germ layers in the body. That is where the organs and other structures are going to develop. Okay, once the completion of your blastocyst takes place, it will indicate the beginning of 3 germ layers or formation of 3 germ layers. Once the formation of 3 germ layer takes place, we are going to study about the formation of different organs and also formation of different structures present in the body. Okay, and this is about IVF. Any idea about IVF? Yes? Yeah. In vitro fertilization. After that, how the in vitro fertilization takes place, what are the basic things we should, yes, outside the, yes, we can do it in, Any other points for IVF? For fertilization, the clinical importance is IVF. If you get it for 10 marks, you have to write about the 3 conditions. That is, the first one is capacitation and acrosomal reaction, then the fusion of male and female pronuclei, or you can write the heading as fusion of male and female gametes also, then followed by results of fertilization. In the clinical part, you should mention about IVF for 10 marks. Okay, so what you are going to write about IVF? Okay, fine. IVF I'll discuss in the next class, okay, once again, because that also includes like step-by-step process. If I discuss that again, you will get confused with the fertilization process and it will become too much for one session. So in the next class, I'm going to discuss about the IVF followed by formation of blastocyst also. Okay, any doubts in the fertilization for today's session? No doubts? Everything is cleared? Sure?